Good morning. My name is Abhinav Raghu. I go to Salem High School, and my mentor was Annie in the Moore Lab. And my project this summer was characterizing the effect of benzene-induced in maternal inflammation on fetal macrophage development. Uh, First, I'd like to start off by talking about air pollution and its effect on human health. So nearly 91% of the world's population breathes in polluted air every day. These compounds travel throughout our body and cause negative impacts on all our body systems, such as asthma, diabetes, stroke, heart disease, to name a few. However, the full effect of air pollution on the reproductive system is unclear. There is recent research that shows that air pollutant can be linked to preterm birth. One of the biggest contributors to air pollution is benzene. Benzene is one of the top 20 most produced chemicals in the US. It's an odorless and colorless gas that easily evaporates in the air. It's very common and has many sources such as volcanoes, wildfires, petroleum, cigarette smoke, car exhaust, and more. Next, I'd like to talk about the modulation of the immune system during pregnancy. So during the first trimester, the immune system is in a mostly inflammatory state to allow for implantation and growth of the placenta. The second trimester is a non-inflammatory state to allow for fetal growth and development. And the third trimester is inflammatory again to allow for labor and delivery. During pregnancy, the placenta is a maternal fetal interface that has many functions, such as providing nutrients like oxygen, removing waste like carbon dioxide and nitrates, and most importantly for my project, it directs the immune education of the fetus, which leads me to the role of immune cells during pregnancy. So previously, it was believed that these cells were suppressed during pregnancy, but research shows that maternal fetal immune cells actually work together to promote immune education. As you can see, there are many immune cells in the placenta, such as NK cells and T cells, and the focus of my project, which are macrophages. So the macrophages in the placenta have many functions, such as the invasion of trophoblasts, vascular remodeling, damage repair, phagocytosis of debris, and they also work to prevent any transplacental inflammation and protect the fetus. All of this leads me to the main aim this summer, which was to characterize the effects of benzene-induced inflammation on maternal fetal outcomes using an in vivo mouse model. And my specific hypothesis for this was that gestational benzene exposure causes abnormal maternal inflammation, leading to dysregulated development and function of fetal macrophages. This is the method of study for the aim. So pregnant mice were exposed to either control or benzene air from embryonic day 0.5 to embryonic day 12.5. The benzene exposed mice were exposed to a concentration of 5 parts per million for 24 hours a day. And at the end of the exposure, the placenta, fetal organs, and maternal serum were collected for study. With all of this was the first research question, which was that how does gestational benzene exposure cause adverse pregnancy outcomes? And in order to study this, we analyzed the litter and fetal characteristics. So first we looked at litter characteristics, and we saw that in both litter size and in implantations, there are no significant differences between control and benzene litters. However, when it came to resorptions, which are a measure of preterm birth, they are significantly increased in benzene-exposed litters. After this, we wanted to analyze the individual fetus. So we looked at fetal characteristics. We used three measures, which were fetal weight, crown to rump length, which is this right here, and occipital frontal diameter, which is this, and found that in all three of these metrics, the benzene-exposed fetuses were decreased. This leads me to the conclusions from question one, which is that gestational benzene exposure does lead to adverse pregnancy outcomes. We saw that the benzene litters had significantly increased resorptions, which are a measure of preterm birth. And we also saw that the benzene exposed offspring had significantly decreased weight, height, and head size, which shows in utero growth restriction. After this, we wanted to analyze the mother. So my research question two, which is what is the maternal inflammatory response to benzene exposure? And in order to answer this question, we performed a test called a Luminex assay to test for inflammatory cytokines. We tested for 6 cytokines, but only 2, RANTES and GCSF, were within readable range. And we found that in both cytokines, there were no significant differences in concentration between control and benzene exposed mice. which led me to the conclusion from question 2, which is that there is no significant inflammatory cytokines measured on the maternal side in response to benzene exposure at 5 parts per million. After this, we wanted to look at the placenta and fetus. So our third research question was, what are the placental and fetal responses to benzene exposure? And in order to analyze this, we performed two experiments. First was a flow cytometry for placental macrophages. We wanted to identify the inflammatory macrophages in the placenta, which were identified as being positive for both CD11B and MHC class 2. This was our gating strategy. So first, we gated for cells from the debris, and from the cell population, we gated for the single cells. From the single cells, we applied the CD11B marker as the general marker for macrophages, just seen right here. And after this, we use the MHC class 2 as a marker for inflammatory macrophages. As you can see here, there are two distinct populations, and the population on top is MHC class 2 positive. After this, we performed an analysis of the potential macrophages and found that benzene female offspring had significantly greater percentage of inflammatory macrophage than benzene male offspring. But when it came to the MHC class 2 negative populations, there were no significant differences between any groups. After this, our second experiment was qPCR. We performed qPCR for 3 genes in the placenta, which were IL-1 beta, IL-7R, and IL-6. And we first looked at the baseline sex differences and found that in both IL-1 beta and IL-7R, there are significantly increased expression in control females and control males, while IL-6 had no significant differences. IL-1 beta and IL-6 are both pro-inflammatory genes, while IL-7R is a gene that is necessary for the differentiation of hematopoietic stem cells. After we looked at the baseline sex differences, we wanted to look at the exposure differences and found that in all 3 genes, the benzene-exposed male offspring had greater expression. While the difference was not significant, the p-value for IL-1 beta was 0.06 and for IL-7R was 0.07, which suggests that we may need more N to confirm this trend. We found that the females had no significant differences. Since we saw that the benzene male offspring had increased expression in the placenta, we wanted to see how the fetus reacts. So we performed qPCR for 4 genes in the fetus, in the fetal brain. The first 3 were the same genes that we performed for in the placenta, and the 4th gene was IRF8, which is a gene that is necessary for the differentiation of macrophages, specifically in the brain. When we looked at the baseline sex differences, we found no significant differences in IL-1 beta, IL-7R, or IL-6, but increased in expression in control male than control female. After this, we looked at the exposure differences and found that all 4 genes, the benzene male offspring showed decreased expression of these genes than their control male offspring. This difference was significant in IL-7R and IRF8. While we didn't see any significant differences in IL-1 beta and IL-6, we decided we may need more N to confirm the trend. The females had no significant differences. This led me to the conclusions from question 3, which is that benzene exposure at 5 parts per million does induce inflammatory changes in the placenta and fetal brain. Second is that there may be an inverse relationship between the placenta and fetal immune response to benzene exposure. And third is that there are differential response to benzene exposure between male and female offspring. With this, I moved on to 3 main next steps to continue this experiment. First would be to perform flow cytometry analysis of macrophages and fetal organs such as liver, lung, and brain to assess the effects of benzene on the developing immune system. Second would be to perform modulation of mechanistic gene targets to link placental immune responses and fetal macrophage development. And third would be an in vitro co-culture of trophoblasts, which are SWAN71, and monocytes, which are THP1 cells, to translate and support in vivo studies. Lastly, I'd like to thank everyone that made this program possible. First, Dr. Moore for making this program, my mentor, Annie, and all the other Repro Group mentors, Anthony, Grace, Candy, and Bean, and also Alyssa for all the help with the administration, and everyone at the Mott Center that agreed to be a mentor for us high schoolers this summer. The funding was provided by an NIH grant. Thank you.